Carl June. So, Stephen.
Thank you, Ed. It's great to be here. It's a terrific annual event, and we're going to start with something totally different. I come from the small molecule world, which is certainly well understood, and now we're moving to a completely different area where we make custom drugs for people on demand to meet their particular circumstances. And this was a totally unorthodox way of thinking not that very long ago. So I'm joined today by Carl June, who's professor of pathology and laboratory medicine at University of Pennsylvania. He heads their cellular immunotherapy program, and he's one of the leaders and pioneers in the field of turning T cells into patients' drugs. So I thought we'd use this time. We have about 45 minutes. I'd like to divide it between sort of where the field is, so everyone is sort of caught up and where the field's going, and then move to the more controversial topics about how do we finance this, what are the public health implications, and how do we manage this. And then I want to leave time, a good 15 minutes for you all to ask questions, because I think that's a really important part of this conference, and is to get everyone engaged and off to a fun start. So, Carl, why don't we start with just generally where you see the field and where you see it going?
Thanks, Steve. And first of all, I want to thank Raj and Ed for organizing this. And I think clearly, I mean, CAR T cells are the ultimate right now in personalized therapy, and it's a disruptive event and requires a lot of changes. So it's right in the theme of this. A little bit of history is that, we first began treating patients with CAR T cells in 1997, and those patients actually had HIV. And we were doing that at that point because there were no integrase inhibitors and so on, and that people died of the immunodeficiency. And we found we could repair the immune system in those patients, and we now have just submitted an abstract of where this is quite remarkable because the FDA's database, where people on a gene transfer protocol, you follow them, for 15 years. And so we followed those patients. The first 18 we treated, and we just submitted an abstract. We have follow-up now from 1997 on 15 out of those 18 patients. They all still have CAR T cells, and they're all well. I mean, they're now on therapy for their HIV, but their immune system is healthy. The CAR T cells have survived for decades, and there's no autoimmunity. There's none of the awful things that could happen with gene manufacturing and genetic engineering. So that's, we then began treating patients with cancer with CAR T cells in 2010. And the initial patients had chronic lymphocytic leukemia that was chemotherapy refractory. All 3 patients responded, and 2 out of those 3 now are disease-free and still have CAR T cells like our HIV patients did after a single infusion. And they have no leukemia. I mean, we have very sensitive, deep sequencing of the bone marrow, and that clone has gone at very deep levels. So I think we can say 2 out of those 3 patients are probably cured 9 years down. Now, the liability they have is they still have ongoing B-cell aplasia because the CAR T cells are still working and still target. They don't tell the difference between a normal B-cell and a leukemic B-cell. We have found now that that's an acquired immunodeficiency. It's more severe in children than adults, because adults have had a lot of infections, and their plasma cells that make the memory antibodies are CD19 negative, so they're spared. And so our adults don't require anything except observation. The children need gamma globulin replacement therapy because they haven't, built up that immune memory. So that there's a lot of room for more innovation. But the idea that you could have a so-called living drug, I think, is now established both in cancer patients, at least with leukemia and lymphoma, and chronic infections. So there's a lot of runway for new kinds of therapies. And so, based on just those 3 patients in 2011, then we reported them, and now they're 9 years on. But back then, we had the first real mechanism of action. We could show correlation between the proliferation of the CAR cells, tumor lysis syndrome, cytokine release syndrome, and then eradication of the leukemia clones. And so Novartis called us up and said, what about commercializing this? And we had never even thought, no one thought it was possible in 2012. And now we have a whole industry around that. I've been told there's more than 50 biotechs right here in the Cambridge, Kendall Square area now doing cell therapies. So it was a disruptive change based on 3 patients. And it's completely changed a lot of our approaches. And probably the biggest issue now scientifically is, can we move beyond hematologic malignancies? And there's a lot of work on that. In animals, you can. Engineered T cells work in solid cancers, but it's more complex, and we can talk a lot about those.
So Carl, the regulatory environment for gene therapy has changed a lot from the early days in the 90s when it first took off to where we are today. How has this evolved, and what are the continuing needs for the regulatory environment to make this commercially doable and accessible to everyone?
Yeah, so that's a really interesting area, Steve. So, I mean, there's been hype and hysteria when you have gene modification. Would there be all kinds of Frankensteins created and so on? And so it's not dissimilar to the Asilomar conference many years ago where they went with DNA cloning. And educated people made different decisions then. I mean, Harvard decided to get out of that back, remember? And then, but other places went ahead and worked on recombinant DNA engineering, and then the fields matured. So we have now the issues of, genetic engineering. We have orthogonally things like CRISPR-Cas9 engineering. The ability to make synthetic immune systems is much more than I would have imagined back in 2010. So it's a huge issue. And there are 2 main things that, we have the regulatory thing, which is the FDA never evolved with the idea of an ultra-personalized therapy and the standards you have in that compared to something that's, a small molecule, which, as you know, is a very different beast than a manufactured single patient N of 1 therapy. So the FDA has been as forthcoming as they can, but the regulations are very different on that. Another thing that's become evident is that for the first time, I think, the drug life cycle of these, basically it's right now, it's an engineered T cell, which has been shown safe in more than 1,000 patients now. We've never had, a genetically modified T cell transform. So that's different than stem cells, where that has happened in some highly publicized trials. So there's a cell type specific safety issue. Post-thymic T cells are very safe, and in fact, you can try to make them turn into a leukemia cell, a T cell leukemia, and it's very difficult. So there is cell type specificity when you do genetic engineering. But the basic backbone now of an engineered T cell has been shown safe. And so there's now all kinds of innovation. And for the first time, I think, in the biopharma industry, patent duration won't matter. It's become more like cell phones and stuff, where Qualcomm can have patents and so on the chips. But new cell phones come out every 2 years if you want to have a competitive product. So we have that now with the cell and gene therapy thing, where new things are coming on in innovation. And so in a pharma, how do you manage that? It's a very different patent management strategy. It's different. And innovation has to be the driver, I think. And then the FDA is not equipped to handle that kind of rapid drug cycle management. So that's going to be a bottleneck, I think. It's very difficult because they never had that kind of turnaround, with registrations and so on. And how do we have, how do we follow patients, get safety data banks, and so on?
So you've talked about the expansion in the field and the number of players that have entered this, perhaps somewhat unexpectedly. The biotech industry, large pharma here in the U.S. and elsewhere, certainly a tremendous amount of work in China. Where do you see this going? Will these diverse sectors harmonize? Will there be some regulatory standards? With so many people doing so many different things, does that produce more opportunity or just more risk?
So I think, hopefully it's driven by science, and the regulatory science needs to evolve. We didn't used to have that, I think. And so there's a, you know, a real dissociation between the amount of people with experience in this field and now that's, so that's a vacuum that's going to be filled by people who get experience. And I mean, it's clearly worldwide. There's more actual trials and companies and investment in China now than there are here in the US. It all started here in the US, but China has, I've just come back from there. It's incredible because it's both government backed and then VC backed. And so we need to really pay attention to that at a national level. You know, China's always been looked at sort of as a Me Too market. And now, for the first time, they're driving innovation, I think. So that's an interesting issue, and can we have worldwide harmonization? You know, last fall, there was that highly publicized event where basically illegally and ethically unsupportable, you know, genome editing was done at a vertical level to engineer fetuses. And, you know, that happened in China. It was done illegally, but we need to make sure that doesn't happen because that could really stymie the whole field. So having harmonization between Europe and Asia and the US is really important.
So to that point, traditionally U.S. medical societies, the AACR and ASCO and ABA and AHA and a whole collection of initials, they've been the sort of guardians of the standards and have driven physician communities toward, you know, best practices. Do you see this happening? Is the right level of engagement there to, not to police it, but more or less to guide the field at the patient level so that we get to best practices?
Yeah, I mean, so that is happening, and it's happening at different rates, for instance, in Europe than it is here. It's hard to know you know, one example is what happened with bone marrow transplantation. So as you know, that started out as an academic. There were never INDs, and it evolved in the academic world, and the FDA did not regulate it. They made a decision back with gene transfer that this would be, engineered cells would be regulated. You know, so if it's more than minimally manipulated, the standard is the FDA requires that it be done under INDs. And so can it go to the level like of Red Cross, you know, blood transfusion medicine, you know, or will it be central pharma manufacturing? And I think over time, I mean, right now the FDA has come down to the position that we require central manufacturing and pharma level control. But I think there's going to come, not in in less than 5 years, we'll have the ability to do point of care in this. And then it's more like a transfusion medicine business where, I mean, at a local hospital, patients can have blood drawn, cells manufactured there, and just reinfused. So that then requires a very different oversight. And if there's comfort by that time, I think that will happen. You know, that there'll be certifications. We need, for instance, validated processes on, you know, transduction of cells. And if you have an error rate that's, you know, sigma, 6 sigma or something, I think that will be acceptable in cancer. So the point of care will really change things. Right now, our manufacturing is done in the industry with the technologies. Basically, my lab started in the 1990s, and it hasn't been advanced. There's many new engineering approaches that could make it safe. You know, the issues of product identity, contamination, and so on, basically eliminated with proper engineering.
So from the small molecule or even recombinant protein side, large companies and biotech companies are obliged to and do well pharmacovigilance, following their products on the market and reporting outcomes, and the labels evolve over time. If this does evolve to a point of care, say your product is made at your local hospital in their blood bank, how do we do pharmacovigilance? How is that outbred system going to inform the public when things go wrong?
So that's a good question. There's been asymmetry in that gene and cell therapy have been overregulated compared to small molecules where there's a bigger history. So it's well known to oncologists that if you get cytotoxic chemotherapy for breast or ovarian cancer, you have a 5 or 10% chance of getting leukemia later, from genotoxicity. And we don't have forward-looking registries on that. It's just, but the cell and gene therapy, we do have that because we had a separate regulation that would follow people. So I think the pharmacovigilance needs to be made standard across all therapies and not just, it's asymmetric like it is now. And I think, so we need to have that ongoing registry, even when it becomes point of care. So that needs to be built into the system. It's really important because the long-term risk really of the kind of engineered cell therapy we're doing right now is not, it's turned out it's not leukemia or genotoxicity, it's probably going to be autoimmunity. And we won't know that long-term risk unless patients are followed.
Let's turn to the issues of pricing, reimbursement, and access. These therapies aren't cheap. They're not cheap to manufacture. There's a lot of logistics on the delivery side. I'm not sure any of the companies are making much money in this business yet. Certainly in talking with hospitals and healthcare organizations, often these are money-losing. How does that change? That's not sustainable if we have multiple products of this type. So what needs to change for this to make this more affordable or more generally accessible without subsidy?
So, you know, it's a complex issue. You need to break it down into the different parts. I mean, one is the actual cost of goods. And right now with the N of 1 therapies with Carty cells, for instance, the main cost driver is human labor. So automation clearly could make a big change in the cost of goods. And I think that's certain to happen. There was no industry before to try to, drive cell therapy efficiency and so on. And it just will evolve just like it has for recombinant proteins. So I think that's a given. But then when the cost of goods goes down, I mean, we have an issue, probably the worst place to open up a new therapy is the United States healthcare system, because it's the fragmentation. And
I'm sorry to hear that.
so, I mean, that's a way above our pay grade thing that needs political adjustment on how to deliver care, pay for performance type things and all those. So once the cell manufacturing issues are worked out, the complex logistics and so on, then it enters the rest of the things, some of which Raj was talking about. What do you do in orphan diseases? How do you set pricing and so on? As an anecdote, I was in Brussels last month where after a talk, one of the physicians came up and said they were going to treat a 25-year-old with leukemia who was actually a medical student the very next day. And so I said, great, this is a Novartis product, which I've worked with them a long time on that. And so I said, Novartis has opened up a manufacturing plant in Leipzig, Germany. I said, is that how you did it? And they said, no. In this case, the patient was apheresed in Brussels, and then they flew the cells to Morris Plains, New Jersey, manufactured, froze them, and sent them back to Brussels. And so that obviously is a very complex logistics. It takes time to scale this out. So it's happening, but we have an unknown time frame of how long it will take.
So before we go to a Q&A session, maybe we could talk about where this field evolves therapeutically. You talked about HIV as where it got its start, and then now it's entirely in cancer and in various embodiments, and solid tumors are certainly big on the horizon. But beyond cancer, what else are T cells good for?
So as you know, I mean, almost every chronic disease has a touch point with the immune system, whether it's dementia, whether it's cardiac disease, and autoimmunity. Maybe 15 or 20% of the population has autoimmunity at some level, and from systemic or organ specific. So the really exciting thing is that engineered cells, T cells and other kinds of cells, can dial down the immune system as well as turn it on. So I think in chronic infections and cancers, I mean, we'll see many, many different advances happen. But there are now several companies and trials underway to dial down the immune system. We've worked with Bruce Blazer at the University of Minnesota and shown that graft versus host disease can be prevented using engineered cord blood regulatory T cells, for instance. And there are trials now looking at diabetes, solid organ transplants, and so on. So I think that will happen. And the holy grail has been, can you get away from daily immunosuppression and have, curative therapies for autoimmune disease, which is, striking that, when I was in medical school, we didn't know the cause of cancer. And then, as we mentioned, we learned how to sequence the genome, and now you can actually engineer, cures. But for autoimmune disease, we don't have that yet. And it's the, huge part of our national health budget. And I hope, I'm sure there's going to be lots of innovation there to get there.
Okay, so let's open it up to the audience. I know we've covered a lot of territory, but I'm sure you have very specific questions you'd like to hear addressed. So we're happy to entertain your questions. While we're waiting for the audience, we've talked about mainly T-cell focused therapies, but that's sort of one niche. Do you foresee that other cells were going to be manipulated ex vivo and transplanted, or will we get to in vivo transduction of cells where it's not manufactured outside the body?
So, I mean, I think that it's really a blue sky thing where all the bone marrow derived cells you can genetically engineer, and you can find vulnerabilities you can correct. I think one of the huge things that will happen will be the cure of sickle cell anemia. We've known which codon needs to be changed for sickle cell anemia, and that technology exists now. And for the first time, instead of a lethal disease that's extraordinarily expensive, we're going to be able to cure that with engineered stem cells. And there's many other examples like that. There will be, natural killer cells, dendritic cells, et cetera. So macrophages. We have one of my graduate students recently just started a company where he makes CAR-Macs. So they're CAR molecules put in macrophages, and then the macrophages, phagocytose, basically eat tumor cells. So T cells kill with cytolytic effects. Macrophages do it by phagocytosis. So there's all kinds of innovation that's going to be very exciting to watch.
Great.
Good morning, John Armstrong from dermatology company Galderma. You were talking about the challenges facing us for applying CAR-T therapy to autoimmune diseases, and I know they're large. There's work done by Amy Payne on the development of a CAR-T therapy for a derm disorder called bullous pemphigoid, where you have to know what the autoantigen is, in this case, desmoglein 3. But for the vast majority, all but that one, we don't know what the autoantigen is. Is there another approach you're thinking for making this therapy applicable to autoimmune disease?
So, I mean, that's a really exciting thing. Don Siegel, who's here, actually made part of those. They're called chimeric autoantigen receptor T cells, so CAAR T cells. And that trial will begin, sometime next year to see if, in that case, bullous pemphigoid, where the known antigen and antibody thing could be interrupted. So that will be kind of a proof of concept. But I think, as we, the famous saying, Richard Feynman said, unless you can take it apart and put it back together again, you can't really understand it. Yeah. And so there's some autoimmune diseases we understand what causes them, like bullous pemphigoid, but there are others we don't. And so once we understand, I think there will be engineering approaches then for most of them. And the fact that you can regrow skin now is a huge thing. So I think many corrective gene therapies will happen, and it's just very exciting to watch that happen now.
Thank you.
Good morning. I'm Stephen Wicks from the Colorado Center for Personalized Medicine. I wanted to ask what kind of regulatory regime you envisage, because I'm kind of scared by the kind of local manufacture. As a former inspector, I've seen some quite scary things, so I just wondered what you think about the kind of thing in the future.
So obviously, I mean, we need control systems where they're validated. So I see it mostly as an engineering problem. I mean, the main issue if you make personalized cells, really it's chain of custody and identification. Because if you give the wrong cells to the wrong patient, they'll likely get a very bad graft versus host disease. So, but we already do that in transfusion medicine. And, two people sign off, et cetera, barcode. There's ways to make it really foolproof. And so that has to happen. But beyond that, I think we can have validated systems So that there's still regulation there, but we need the systems that are being built but don't yet exist.
Do you see it as a light touch or more of a kind of regulated inspection?
Well, I think, what I didn't mention is there's kind of two ways the field's going. One's going to be with, autologous cells, which have a lot of advantage of safety and, they can last a long time and not do mischief in a patient. At least they haven't so far. And then the other approach now is third-party cells, where it's much more in a pharma model, and they could be engineered from cord blood or iPSCs. And then those will be off the shelf and be frozen and probably sitting in pharmacies ready to go. So it's the ultra-personalized ones will really benefit from local manufacturing, and we need to see what role the off-the-shelf kinds of engineered cells have.
Thank you.
So you referenced the current blood banking procedures as evidence that this could go forward safely, but there's a fundamental difference between blood banking and what we're headed toward. Red cell transfusion is a pretty vanilla topic. I mean, it's the same thing over and over. And here you're talking about you're going to, in principle, be doing one thing infrequently. I mean, you're not going to see an ALL patient every day. So how do you maintain standards when you have such a diverse portfolio of potential products? Any one hospital, take Lancaster General in your backyard, they're not, they're sort of an academic center, they're affiliated with you, they know how to deliver good care. But they're going to have a very outbred patient population, and if we're treating skin disorders and cancers and HIV on any one day or any one week, they're not going to be doing much of anything twice, and that would seem to be a good opportunity for mishap.
Yeah. So, I mean, you can't gloss over the history of, red blood cell transfusion initially wasn't so routine until Landsteiner figured out how to do blood typing. But so it was initially not a good thing a lot of times. So I think we're at that level where it's evolving, and so you need to get trained people. There's always going to be this inverse zip code rule where if you're close to innovative centers, there'll be better outcomes and people who are trained and know how to do things. Whereas out in rural America, there's a whole generational problem, I think. So implementing it there is very different than here in Kendall Square or something. So, so that's a type 2 translational issue of how to move it out into rural areas, where I think you're right. I mean, it's much more of a problem. But I mean, right now it's, acute leukemia, but just in a year from now, probably it's going to be myeloma. The most common heme malignancy will be FDA approved with Carty cells. And so that's frequent, and places need to learn how to do that if it's going to be delivered efficiently. And so we have an issue it's really across all of healthcare of how to have innovative therapies move into the community. That's not unique to cell and gene therapy.
Zen.
Yes. Hi, Jen Mahoney from Asuragen. I wonder if you could comment on what kind of diagnostic innovation needs to happen to move this whole ecosystem forward.
So it's, I didn't really mention, but it's kind of like hand in glove. For cancer going forward, it's not just, cancer genome sequencing and epigenetics need to be known, but the whole immune system needs to be bookmarked and tested in patients at diagnosis, because that is going to be used in designing the right therapy for a given patient. So this is an evolving area of diagnostics that didn't exist before, whether it's liquid biopsies, whether it's genomics, epigenomics, but both not just in the case of a cancer, but as well as the patient's immune system. So we found you can predict very highly in chronic lymphocytic leukemia at the time you draw blood from them, who you can actually successfully manufacture CAR cells and who will be cured and who won't be. So then you could avoid an expensive manufacturing and futile effort and go maybe straight to some other kind of therapy, maybe with third-party cells. So that's an evolving area, and we need that space, as well as cell manufacturing and then the science and engineering.
Thank you.
One area we haven't talked about is product liability, and I know you have some views on this. Certainly for your typical small molecule or biologic, companies carry a fair amount of liability risk from either real or perceived injury. How do you manage liability on a product that has only been made once?
Class action lawsuits actually started in Philadelphia. And the term Philadelphia lawyer happened, and it all was actually because when the polio vaccine came out. And Paul Offit's written some really great books about that. What happened was they had recombinant polio virus. It was not fully attenuated, and some kids getting a polio vaccine got neurotropic polio and ended up on iron lungs. So that was a huge class action lawsuit. And the first examples of those. The actual unforeseen benefit of an ultra-personalized cell therapy is there's no class action lawsuit. So it's N of one. So to that extent, there can be lower liability. I mean, now you can always have some problem, like if you have a recombinant lentivirus or whatever that has recombinant, it's not error-proof that way. But I think this is one area that's unforeseen where it's probably lower liability for the pharma industry.
You wrote an editorial recently, which we chatted about, on a publication that came out of some research done in China. And I think one of your comments in the editorial was, the most important thing about this paper is what it doesn't say. Would you care to comment on that, or just maybe describe the paper a little bit and what the issues that pointed out?
Yeah, so that was a single case study of a young 23-year-old in China who had two sequential diagnoses, maybe 3 months apart, one being acute leukemia and then HIV/AIDS. And so they used that as an opportunity to try to replicate the Berlin patient, where 10 years ago, a patient in Berlin had acute leukemia and then got a transplant from a cord blood patient who was identical in the chromosome 6 area for HLA, but also had on another chromosome a very unusual thing, which is about 5% of Caucasians had. It had the null delta 32 mutation of CCR5, which means you cannot infect it with HIV. So they gave that patient a transplant with that, and were able to then, he's the first patient to be said cured. We have a second patient this year where that just reported in Nature now, where because of this fortuitous outcome of using a genetically matched product that's also naturally HIV resistant, the patient then eradicated that. So they did this successfully in China, where they tried to engineer using CRISPR-Cas9 technology the delta 32 mutation. So they found an HLA-matched cord blood donor and then knocked out CCR5, did the transplant, and got nice reconstitution, but it was only about 8%. The efficiency was not high enough, and the patient still has HIV. And his leukemia went away. So the issue there was not enough feasibility, the manufacturing part was not enough to really test that hypothesis. But that's just an example of things that could be done. I mentioned sickle cell anemia, where we're going to be able to fix that with, I think, autologous cells. And so it's very exciting, the fact that you can now, engineer a stem cell and then have it regraft, and then all the lineages came out of that. So that's shown now for years follow-up, and no side effects. And the big issue's been, are there off-target edits and so on, and they didn't find them.
In an N of one.
N of one. Yep. Yeah.
Stephen. One more question.
One more, okay. Yeah. One more question. So you've described the, I'm looking at the clock down here, so your clock doesn't
The
match.
clocks are not synced.
Oh, thank you. I like that clock. So, we talked about the affordable issues and the profitability issues now, and then you described this wonderful future state where you can get it at your corner hospital. How do you get from A to B? That's a very nice place to be in the future, but it's not clear how you fund that gap.
Yeah, so one thing that's really clear is that it depends on what society you're in. So Europe is very cautious in this area, and they're way behind. if you look at clinicaltrials.gov and the activity here. And it's going to take longer there. I mean, China is ahead of us, and I mean, it's like the Wild West. And it's
Wild East, I think they're east.
Wild East, I guess. So, but the fact is, we'll probably learn from them. And I think we have more of a science-driven approach. And so getting from A to B is going to be a set of, you walk before you run. And it will happen at leading medical centers and then be translated into the community. So how you fund it is a very different issue now. I mean, it's how do you integrate it into our healthcare system, as you mentioned in the beginning, is probably the hardest question now.
Okay.
So,
Let's stop here and
I was going to say, please join me in thanking Carl and Stephen.